To the MRL for ivermectin for sheep, pig, and goat tissues, and that is right now held at step 4 of the Codex procedure. And I guess, Mark, the floor is yours to begin with.
Okay, thank you, Samuel. As indicated, the CCRVDF at their last meeting had requested that GFoRSS provide recommendations for MRLs for these particular species and tissues. And they're available for comment at step 3 and essentially will be discussed at step 4 at this upcoming meeting. So ivermectin is not a new drug. It's been used in human medicine probably for the past 40 years to you see there's a variety of diseases primarily caused by parasitic roundworms and nematodes that affect humans. So there's a good history of safe use of this drug. GFoRSS and Codex have looked at developing MRLs for ivermectin in other species, primarily cattle, and that goes back to as late as 1993. So there are other MRLs available for ivermectin. There's a bit of a history of the toxicological evaluation that GFoRSS has considered. We haven't gone into great details of it, but simply at their 81st meeting in 2015, they established a new ADI. There was a previous ADI of 1 microgram per kilogram body weight per day, which is a little more conservative. But it actually makes a good case study because there was an issue with the test species used for the ADI that was established in 1993, which GFoRSS decided was overly sensitive and was not representative of likely human responses to ivermectin. So usually ADIs go down or become more conservative. In this case, they decided that the previous ADI was too conservative, and they actually increased it. So, and at the same time, an acute oral reference dose was recommended by GFoRSS, and that was at a level higher than the ADI, which isn't unusual. So, and as typical is the process, GFoRSS has begun to try to recommend acute oral reference doses based on human data when available. And in this particular case, there were human studies available from ivermectin's long use as a therapeutic drug in humans. So Samuel has listed the MRLs that were recommended following the 81st meeting, and they're relatively low. So, and the 88th meeting in reviewing the existing MRLs provided a bit of description in terms of using MRLs for other similar test species and applying those to the species at hand. And this is mainly done when there's a lack of residue depletion studies. And the committee is relatively certain that the extrapolation species is similar enough in metabolism and disposition of the drug at hand to be able to allow for this extrapolation. There were estimates provided both for chronic daily exposure, and because there's an acute oral reference dose, acute daily exposures would also be considered. And in typical GFoRSS fashion for vet drugs, this was done both for the general population and for children. So you see that this makes a minor contribution to the ADI. The chronic intake estimations are between 4 and 5.9% of the ADI. So and which is typical of the acute oral reference dose. In certain cases, there was no information for consumption available for certain of the tissues and species. So in this particular case, GFoRSS went with the most usually and highly consumed in terms of amount for the acute oral reference dose intakes. And in this particular case, the acute intake for children was based on only consumption of cattle muscle because that was the commodity with the highest intake for children. And this was, you see that it only accounts for 41% of the acute oral reference dose, is fairly high. But then the value that they used for sheep muscle was only 0.5% of the acute oral reference dose. So simply all they've done is taken the intakes for the particular tissues that are available, applied the MRLs or the recommended MRLs in developing these intakes. And there is an approved analytical methodology for ivermectin, and this is based on LCMS-MS. And it also refers to the fact that there was previous MRLs established for various other tissues, including more recently cattle muscle. So you have a variety of previous MRLs that have been established. So essentially, because this has a long history of safe use, there are other countries that do have MRLs. And the issue with, in general, with GFoRSS and Codex, these MRLs are typically on a requested basis. So when you see the MRLs listed for Canada and the US, this has generally been requested by the proponents or the people that are sponsoring the application of this particular use. It's only more recently that the EU has gone to, when there's broad use of various veterinary drugs, they have assigned the MRLs to all mammalian food producing species. So in this particular case, you can compare the ivermectin MRLs that are currently in use in the EU. And this is for fat, kidney, liver, and muscle. And it's applied to all mammalian food producing species without identification. So we've put a small possible path forward, including the fact that there will likely be support, considering there's broad use of ivermectin in veterinary medicine and in human medicine. And the issue would be is that there probably will be support for moving the MRLs to step 5, but not for the finalization step 5.8. And this is basically due to the fact that some delegates have noted that some of the MRLs being recommended for these species are relatively low and incorporate a substantial safety margin. So it's sort of the reverse situation. Typically, delegates would complain if the MRLs are too high, but in this particular case, they're sort of questioning whether GFoRSS is being overly conservative. And in most cases, the use of vet drugs is based on usual practice, which is usually referred to as good practice in the use of veterinary drugs or good veterinary practice. So essentially, there are established protocols for using all vet drugs, which includes both dosing and withdrawal periods. So there have been some questions on whether or not, the information that GFoRSS may be using is different than other jurisdictions for establishing these particular MRLs. GFoRSS did look at additional data when recommending these MRLs, and there's a specific process that they use in terms of looking at these depletion studies obtained from in use of the vet drug. So currently it's expected that because there really is no safety concern, there will be support in moving this at least to step 5, but there could be discussion about the applicability to the JECFA recommended MRLs to other jurisdictions where ivermectin is already approved.
Thank you, Mark, for this overview. So I guess we're going to move to the input that you may have on this. And I see that Dr. Abdel Fattah has raised his hand. I know that Fadi has provided also input in writing, and I'll give you the floor subsequently, Fadi, for that. So, Dr. Abdel Fattah, please.
Okay, thank you. Thank you for this analysis and very nice presentation. I just have a comment about we have to consider the new event about the direct consumption of the veterinary ivermectin drug in human being, not as a residue, direct condition. You see it during the COVID event, COVID-19 event, the practitioner or the human doctor prescribe ivermectin as 4 tablet or 6 tablet per week and repeated by other week with 1 week apartment. So we have to consider the safety of the ivermectin in human being using this overdose, 4 or 6 tablet per week. So this is a direct consumption for ivermectin by human being, not as traces of residues that we are talking about. I see that the European recommendation about to use MRL for all food consumption or all food producing animal, I think is not rational because the difference between the species of animal, and I think the Canadian or the USA studies is more precise because it used different studies for sheep and different studies for swine or pig. And finally, I hope that different studies for camel and the goat to be performed. Thank you.
Thank you, Dr. Abdel Fattah. I guess I'm going to give you, Mark, the opportunity maybe to respond. But I guess just wanted to ask if my assumption is right. Given the low level of residue that will be imposed or that is being factored in here in the exposure, I guess actually if a person is taking ivermectin for medicinal purposes, the contribution of the residue would be somewhat minor, right, Mark? So essentially, I guess it would not have an impact based on this. But I don't know. I mean, this would be my my assumption, but I'm gonna ask you to comment on that.
Thank you, Samuel. No, it's a very good comment, and there have been reports of medicinal use related to COVID-19 for ivermectin. My understanding is that the clinical evidence is not very substantial in terms of it providing a medicinal benefit. So I would not expect that there is a general acceptance for the use of ivermectin in treating COVID-19, but it is a good point because it does relate to potential other uses not considered for veterinary drugs. Now, the good thing about ivermectin is that it does have a relatively low toxicity potential in humans, and that particularly is related to its mechanism of action. So it's sort of an agent that acts on nervous tissue in invertebrates, primarily these nematodes and roundworms, and it has relatively poor bioavailability in humans, mainly likely related to its large molecular weight. So this idea of using it to treat COVID and having relatively large oral doses, it would primarily be related to maintaining a relatively high blood level of ivermectin in order to potentially impact virus, which requires significantly higher doses than are used in veterinary drugs. So as Samuel has described, if you look at some of the intake recommended for ivermectin in human medicine, it's up in the range of milligrams, whereas the ADI that has been established for veterinary drugs is in microgram per kilogram. So there is sort of a substantial difference in human medicine requiring significantly higher doses than in veterinary medicine. So it would be expected that the overall animal use for this, so food producing animal use, would be relatively minor. The point about extrapolation of other species is good. I'm not entirely sure why the EU has decided to relate their MRLs to all food producing animals. But once again, this is considered, I think, on a drug or vet drug by vet drug basis. So in newer MRL deliberations that you see with the European Commission, they are for all mammalian food producing species, whereas for older MRLs, they're more specific, as you see in Canada and the US. So, so hopefully I've addressed your questions.
So thank you for that, Mark. I'm going to move to maybe Dr. Fadi, because Dr. Fadi provided some input in writing, and then I'm going to move to Sami subsequently, to Dr. Sami.
Thank you, Dr. Samuel. Do you want to share the comments, or shall I share it?
I'm gonna leave you maybe the leeway to share. I'm just gonna give you the opportunity to do so. So you are at liberty of sharing your screen, Fadi.
Thank you. In fact, in general, I really found the product on the document is really a piece of work, and it is really putting all the information needed. However, I just tried to add more ideas and comment to support what is really already mentioned in the document. So the first point, the dosage of 200 microgram per kilo is the taken dose in a human. That's ensure the safety of the drug for the human. And the tablet is 3 milligrams. So that's really, in general, is really ensuring that it is safe in general. But the concern, when the drug is used in a human and used in animal, you want to avoid the resistance in the future, which is the same situation if we are speaking about antimicrobial resistance. So in this particular area, we start to think to which level we can decrease the level in the food so that we can't produce resistance in the future. One of the things that I really highlighted in yellow to just for question, because the novel what has been really found in the technical report from WHO 2016 was 0.4 milligram per kilo. As NGFA website, external website, it's 0.5. And the one which is 1.5 milligram, I could not really understand the source of that. And maybe doctor can help us on to explain for us how we can have tuna well. Of course, the acute reference dose is really linked with the 200 microgram taken in tablets, and I think this is what exactly what is really for a chronic or acute dose. But to me, I was not really very understanding this point.
Sure.
Because it is the no effect level. And this is the other point I need to hear. This is the, this is just only the report reference. And the lowell is 0.8 milligram. I just add this information.
Value.
And always I've been asked why we have difference between Noel and Lowell, and always I raise a question on that. Is it really related to the interspecies variation? If there is interspecies variation high, so we will have more gap between Lowell and Noel, or more range, and this range can be decreased if the interspecies is low. It's just, interspecies or intraspecies, but no, this is normally related to interspecies, Lowell and Noel. So this is just can help that 0.8 milligram is really high, considerable of the acceptable daily intake if we need to reach to that level. The other comment is in MENA region, we are really consuming, the ivermectin withdrawal period is 21 days to 28 days. And based on the practice, veterinary practice here, we don't have always really a very high commitment to the withdrawal period. So this can really, in the exposure assessment in the area can be really high. Taking in consideration, it is really being used in massive way. It is not really, the use of the drug is really high, especially for the ivermectin. This is just a comment in the area, and always we keep this in our mind to have a very, more than 10-folds if in the risk assessment situation. The other point related to the liver MRL in cattle, which is 800. This is really too high, and I think if we really have commitment to the withdrawal period, we can achieve below this. So my question was going in my head, why we do have to accept this 800, or why GFoRSS accepted this, taking in consideration we can achieve below in this. And the concept, if the best practice can achieve low level, then why do the MRL can go for high levels? This is really very logical. If we commit with 30 days in cattle, well, it is 28. If we can go 28 withdrawal period, still it is 800. So do we are really, bias the good veterinary practice by putting this number? This is, maybe need technical studies, and I was not really having a time to have more literature review on this point particularly. But I highlighted in yellow for the discussion of today, and I think it is really achievable, and it is one of the concepts in the risk assessment and in the risk management decision that it is achievable. And we have applied this concept for many food additives. It is safe, but we can achieve low level. So we will go for level in our risk management decision in deciding on the MRL. And this was a concern here if we can really achieve this. This is for the 6.1 comments, and I hope it added value for the discussion. Thank you.
Thanks very much, Fadi. That definitely was useful input, and I think I'm going to give the floor back to Mark to address address them point by point, given that we had the benefit of receiving them earlier. So.
Thank you, Samuel, and thank you, Fadi. These are all excellent points. In terms of your first point, yes. I mean, there is a long history of use. Ivermectin has been in use as a human drug to treat these parasitic roundworm nematode infections for over 40 years. The dosing generally is taken orally, and the tablets are typically 1.5 to 3 milligrams, as you've indicated. And so this 200 microgram per kilogram body weight is usually taken over the period of a couple of months. And once again, this is in order to sufficiently clear the parasitic nematodes from the infected individual. So, compared to, this is an effect dose, and essentially as with any human drug, you will have differences in sensitivity. The good thing about ivermectin is, it does have a relatively high molecular weight, so the permeability is limited in humans. This is typically why you have to take a relatively high dose. And essentially, you will end up with, with continual exposure, seeing some adverse effects that have been reported in humans. So, but this is mainly, I think, related to more background information about the use of ivermectin in human medicine. And typically that, other than controlled studies where doses are only given for acute periods of time, is not considered in the GFoRSS assessments. So in this particular case, you have highlighted the fact that the revised ADI that GFoRSS set is based on a NOAEL of 0.5 milligram per kilogram body weight, and that's from a 14-week dog study. So it is the study that you have indicated was cited in the 81st GFoRSS report and the technical report series that you have identified of 997. However, and this is sort of the benefit of being at a GFoRSS meeting, this study, which identified actually a lower NOAEL, was questioned by the committee during the meeting, and they found that there were questions about the conduct of the study, so whether or not it was related to good laboratory practice, and in the interpretation of some of the findings, because I think there was some involvement of the spleen, and which had never been reported in other test species at higher doses. So this particular study that you cited, which has a lower NOAEL, was recognized, but the decision was to go with the 14-week dog study because they felt it was a better conducted and had less questions about the interpretations of the studies. Now, for the acute oral reference dose, this is acute intakes are specifically meant to cover only exposures on a day basis. So this is why the NOAEL of 1.5 milligram per kilogram body weight is higher than the dose that you have suggested for the 200 microgram per kilogram body weight, because the 200 microgram per kilogram body weight, even though it's taken acutely as one dose, that's followed up for a number of weeks, up to 3 or 4 months, or how long it takes to effectively control the parasitic infection. The acute oral reference dose is only for a day's exposure. So in that particular study, and this is part of the human drug acceptability for ivermectin, there were studies with a limited number of human subjects, only 12, that were exposed to increasingly higher doses, only acute, a single dose of ivermectin. And there was a number of factors considered in terms of the tolerability. And it was decided in this particular study that no effects, no adverse effects were observed with a single acute dose of 1.5 milligram per kilogram body weight. So that was taken as the basis to develop the acute oral reference dose. So once again, your point about the liver value specifically for goats, there's a specific process that's used by GFoRSS in establishing MRLs, and this also relates to the your comment about the 800 microgram per kilogram being too high for liver. So essentially what GFoRSS considers, and this is the responsibility of whoever is requesting GFoRSS develop MRLs for a particular vet drug, they have to do these, it's called terminal depletion studies. So essentially, under good veterinary practice, they would administer the drug and then look at terminal depletion. So that means removal by the animal of all detectable residues in all detectable tissues. And if you look at the detailed reports from previous JECFA vet drug assessments, you'll see these log linear transformations of terminal depletion studies. And it's from there where they take the MRL description as being the 95th percentile of the value associated with these terminal studies. So it's a relatively detailed analysis that's done by a statistical approved data package that JECFA uses, and it's consistent with other jurisdictions in looking at how to develop MRLs based on these terminal depletion studies. But essentially, it involves providing the vet drugs to the test species under consideration, and then looking at removal of the residues, so depletion, down to the point where there is none detected. And then they will do a statistical analysis of all that data and plot out the, they call it the 95th, and that's the basically the upper one-sided tolerance, the 95th percentile, of the highest percentages, either a 95th or a 99th percentile of the residues that have been analyzed from these depletion studies. So anytime you see an MRL recommended by JECFA, this is the typical process, unless they specify that oral depletion studies are not required. And then that way they will typically default to providing a value that is some factor of the limit of quantification by the approved analytical method. So this is it's a very slight description of how vet drugs are handled by JECFA, but typically it considers and once again, ivermectin has been approved for use for a number of years in veterinary drugs. So there is a defined good veterinary practice for the use of ivermectin. And obviously it's the lowest dose that shows effectiveness in treated animals for control of the parasitic infections. And it's those doses and the depletion that they rely on to generate the proposed MRLs.
And Mark, can you please just comment on whether, upon essentially deriving an MRL of this nature, and in this case, as highlighted by Fadi, at reaching 800 ppb, basically, is there a concern that if we find that with such an established MRL, we reach up to 50% or above 50% of an acute reference dose? So essentially, it's more a comment on when we calculate the intake with the proposed MRL, and if we find these levels of exposure vis-a-vis an acute reference dose particularly, when do we get concerned, if at all?
That's also a good point, Samuel. Even for acute intakes, the strict summation of the estimated exposure based on the MRL and the consumption of the tissue in question always has to be below either the acute oral reference dose or the ADI. So it would be expected at the time that JECFA established the cattle tissue MRLs that even a value of 800 microgram per kilogram being a significant percentage of the acute oral reference dose, the consumption of the other tissues at the MRLs would not result in exceedance of the acute oral reference dose. If it actually did, then you would have to go back and revisit the MRLs to ensure that the health-based guidance value is not being exceeded. So this is one of the issues that typically you have to follow from Codex meeting to GFoRSS meeting, etc. And as we indicated, GFoRSS at one time did have a significantly lower, an order of magnitude ADI of 1 microgram per kilogram body weight per day. And that's where the original, and this goes back a number of years, that I believe that the original MRL for ivermectin in cattle liver was initially proposed at 100 microgram per kilogram. But that was based on an ADI of only 1 microgram per kilogram body weight per day. So the equivalency of that almost lines up in the fact that the ADI has increased by an order of magnitude, and the MRL that was originally recommended for liver of 100 microgram per kilogram has also increased by almost an order of magnitude. So it's not unexpected to see this high of an MRL, but based on Sammy's point, the MRL assigned with the intake of all these tissues that are used as default by GFoRSS, the summation of exposure would always have to be less than the health-based guidance value.
Doctor, if we are really looking to the EU levels and liver, it is really very low in comparison to Codex. So what is really the justification from EU, and can we consider this as trade barriers? Is there any concerns of developing resistance for the drug in a human in the future?
I think based on the likelihood that some of the intake values that they use for these tissues are relatively high, and it's expected that if you're consuming the amount of liver that they use as a default intake value, you would not be consuming subsequently all the other tissues. I mean, the consumption on a daily basis figure that they use for muscle is 300 grams a day. Yes. So it's expected that if you are consuming liver and kidney, you would not be consuming fat and muscle at their recommended intakes. So how the EU has established the lower MRL.
100. It's 100 basically.
100.
Yeah.
Yes.
Yeah, it's 100.
So I think the EU assessment has the Center for Veterinary Medicine Products has revisited the ivermectin, and that was after GFoRSS had considered that there was some species issues with the mouse species that was originally used to establish the 1 microgram per kilogram body weight ADI was overtly sensitive and was not representative of potential human toxicity. I think the EU did consider that and also accepted the 10 microgram per kilogram body weight per day revised ADI that GFoRSS established.
Okay.
Now, the only thing that I could suggest is that possibly the GFoRSS value has considered different depletion studies in setting their high liver values. But whereas this would be a point for discussion in terms of comparability of MRLs, it would not necessarily be the basis to totally disagree on the advancement. So this is part of the rationale where we have thought that there will likely be support for advancement, but this is only the step 5, so there's additional discussion involved, and typically that will look at potential harmonization of available MRLs across the world in order not to create trade barriers.
Sammy, please.
Okay, thank you, Samuel. The point just is, yes, we have scientific justification to propose draft MRL for sheep and goats, But just a remark, this is those MRL have been proposed based on the new approach, have been adopted in 2018. But Codex have already other MRLs for bovine that have been adopted in 2017. So if we adopt the proposed MRL, we will have in the same table for ivermectin for let's say the MRL will be for bovine in kidney 800 ppb, and for ovine it will be 15 ppb. So what will be the scientific justification for any common consumer that will see the table? It will be assumed that it will be very dangerous to use ivermectin in sheep, and it will be very safe. But we know that it's only a matter of methodology. So the question is, now we will have a new situation in Codex where we will propose an MRL for a tissue for some species, but we already have, let's say, the half of MRL adopted based on the old approach. So maybe the GFoRSS need to reassess data also for bovine in order to have a consistency between different species, because ovine and bovine are not so different. They are not the same species, but they are related species, and we accept to do extrapolation between bovine and ovine. So this is the point. If we go ahead with the adoption of this new MRL, we will have a consistency problem between the different species. So this is my point. Thank you.
And I guess I think actually it's a good point, Sami, because it joins essentially the discussions that we were having just right now. I guess the numbers that we were looking at earlier here were in fact established previously. So these were already, like, the numbers for cattle were already I guess adopted. So the numbers before us for adoption are those ones. And
yes,
for sheep, pigs, and goats, basically. And I guess the question will be potentially to request, a confirmation of the consistency of the approach with previously established MRLs. That's essentially what I'm hearing.
Yes. Yes.
Okay.
For the same compound, we cannot use the new approach for half of species and the old approach for other species. So this is the point. Thank you.
And actually, Fadi, I have your notes already, so I will take whatever extracts from your notes to amend the material that is here as well. Any additional points in relation to ivermectin? VM, I see your hand, so please.
Yes, thank you for this discussion. I have a question about LMRs for ivermectin. Is not there are no studies for camelids and horses?
Mark, do you have the answer to that? Please.
Well, camel and horses, I don't believe have been considered by GFoRSS in terms of developing MRLs. Once again, this would have to be some proponent would have to suggest that they want to use ivermectin in camel and horses in order for GFoRSS to consider that, and then look at either the availability of residue depletion studies, or at least provide evidence that camel and horses are substantially equivalent to a species where MRLs have already been established, and they would have to provide some scientific evidence for that. Now, presumably, ivermectin use in camel and horses would already be covered by the EU, because if these are food-producing animals, the EU MRLs for ivermectin are extended across all food-producing animals. So even though the international food regulatory bodies, whether it's in North America or South America or Africa, Middle East, or Europe strive for a consistency in approaches, it's not always guaranteed that there is. So in this particular case, GFoRSS is looking at an extrapolation potential. Once again, I think they have a working group that's investigating this. If you have developed MRLs for one species, can they be applied to different species? But currently, that is not part of the GFoRSS process. Even though this particular example of ivermectin you see in some of the species, the sheep, pigs, and goats, residue data were not available, and GFoRSS has already suggested that the MRLs for the one species could be applied to the other species based on a similarity. So in the Codex process, succinctly, there are no current MRLs for camel and horses. However, those two species would be covered by the current EU MRLs.